Good day, everybody, and welcome to the lecture of proteins. Proteins are the most fundamental constituents of living matter. They are composed of carbon, hydrogen, and oxygen, like fats and carbohydrates, plus nitrogen. Amino acids are the structural units of proteins. The radical or the side group or the R group distinguishes 20 different amino acids from each other. The amino acids are classified into two types. Essential amino acids are those amino acids that the body cannot build or builds in insufficient amounts and needs to be provided in the diet. And the other class are the non-essential amino acids. Amino acids can be combined together to form a longer chain of peptides. In this diagram, we combine two amino acids and with the liberation of water, forming a dipeptide. And the bond which binds the two amino acids is called a peptide bond. This is the diagram of hemoglobin. Hemoglobin is formed of heme, which is formed of a pigment and an iron atom, and globin. Globin is a protein which is formed of a polypeptide chain. Sickle cell disease. Sickle cell disease, in which the red blood cells transform into the sickle shape. This is due to an abnormal hemoglobin, and the abnormality in the hemoglobin is only one amino acid change or variation from the normal hemoglobin. Protein synthesis. Protein synthesis starts in the nucleus of the protein forming cells. In the DNA, where the DNA forms a copy of itself in the form of a messenger RNA, and this messenger RNA leaves the nucleus, goes to the cytoplasm, to the organelles forming proteins, the ribosome. And the ribosome is formed of ribosomal RNA. The ribosomes start to read the message on the messenger RNA, and it calls the transfer RNAs containing the needed amino acid. And then the ribosomes will combine the amino acids together by a peptide bond till they form the polypeptide chain of the needed protein according to the message present on the messenger RNA. Functions of protein. 1. Growth and replacement. Synthesis and replacement of all tissue needs both essential and non-essential amino acids. And of course, the essential amino acids must be provided in diet. Number 2, immunity. Antibodies or immunoglobulins are proteins. Number 3, fluid balance. Proteins create and are responsible for the osmotic pressure inside and outside the cell that help in maintaining the water equilibrium between the different compartments, between the extracellular, intracellular compartments. Albumin is important in maintaining the normal osmotic balance between the body fluids, as it cannot cross the blood vessels. It remains intravascular. Sodium and potassium balance. Normally the sodium is more concentrated extracellular and potassium is more concentrated intracellular. And this is brought about by the aid of the sodium-potassium pump, which are proteins that maintain their levels, the sodium and potassium, inside and outside the cells. 5. Buffering action. Proteins help to maintain the normal pH of the blood between 7.3 to 7.4 because it can combine with acidic and basic substances to correct acidosis and alkalosis, respectively. Number 6, they provide energy. 1 gram of proteins provide 4 kilocalories. Number 7, all enzymes and many hormones are proteins. And this the table shows an example of some protein hormones like growth hormone, insulin and glucagon, thyroxine, calcitonin and parathormone, and the antidiuretic hormone. All these are proteins hormones. The interaction of the proteins in the diet with the digestive system. In the mouth, there are no enzymes to act on the proteins. but only they are mixed with saliva and crushed with the teeth in order to facilitate swallowing. In the stomach, we have the pepsinogen, and pepsinogen is the inactive form of pepsin. When pepsinogen is exposed to the acidic hydrochloric acid, it becomes activated to the protein pepsin, and this pepsin breaks down protein into smaller polypeptides. In the small intestine, the proteins are exposed to the pancreatic and intestinal proteases that break them down into tripeptides, dipeptides, and individual amino acids. Then finally, the enzymes on the surface of the small intestine hydrolyze these peptides, and the cells can absorb only amino acids after complete digestion. Examples of the enzymes that act on proteins: we have pepsin in the stomach, and in the small intestine we have the enteropeptidase, we have trypsin, we have chymotrypsin, carboxypeptidase, elastase and collagenase, intestinal tripeptidases, and intestinal aminopeptidases. Catabolism of amino acids. Amino acids are catabolized primarily in the liver. In the liver, the amino group is removed by a process called deamination. The result of this process is the production of ammonia, which is very toxic and is quickly converted to urea. All this occurs in the liver. Urea circulates in the blood and then goes to the kidney to be excreted in urine. The rest of the amino acid molecule may be oxidized to release energy if there is insufficient carbohydrates or fats to provide that energy. And if energy is not needed, these amino acids are converted to fats and stored in the adipose tissue. The recommended daily allowance. Infants and children need more protein per body weight than other age groups, since they are growing rapidly and producing new tissues. In addition to pregnant and lactating women, who also need more protein intake. But generally, the RDA for proteins is 0.8 grams per kilogram per day and should provide 10 to 35% of the energy intake. Protein quality measurements. We have 6 measurements that can classify proteins according to their quality. First is the reference protein, which is one of the most complete and digestible protein. And this is the egg protein. Thus, it is used by the scientists as a standard for measuring protein quality of other food proteins. It is assigned a value of 100. Amino acid scoring. This evaluates the food protein's quality by determining its amino acid composition and comparing it with that of the reference protein, which is the egg protein. If the food protein amino acid is 70% of the amount found in the reference protein, then this protein is given a score of 70. The biological value, it measures the protein efficiency in supporting the body's needs. Like the egg protein has a biological value of 100, indicating that 100% of the nitrogen in the egg protein is absorbed and retained. The net protein utilization, NPU, like the biological value, the net protein utilization measures how efficiently a protein is used by the body. The difference is that the net protein utilization measures the retention of food nitrogen consumed rather than the food nitrogen absorbed, which is the thing measured in the biological value. Protein efficiency ratio. This measures the weight gain of a growing animal and compares it to the animal's protein intake. Finally, the protein digestibility corrected amino acid score. It compares the amino acid contents of a protein with preschool children amino acid requirements and corrects the digestibility. This comparison should reveal the most limiting amino acid. This table shows the protein digestibility corrected amino acid score values of selected foods. We can notice that the best, the most complete proteins are the casein, which is the milk protein, which has a PDCAAS value of 1, and the egg white. The soybean has 0.99, the beef 0.92, and the other proteins has got protein digestibility corrected amino acid scoring, which is descending order going down. Protein energy malnutrition occurs in two forms. Protein calorie malnutrition results two similar but distinct diseases. The first one is marasmus, and the second one is kwashiorkor. Both of them are due to protein deficiency. The major differentiating factor between marasmus and kwashiorkor is edema. Marasmus, there is no detectable edema, while in kwashiorkor there is edema. And this table shows a comparison of the features of kwashiorkor and marasmus. This is the edema in kwashiorkor. And this is a picture of marasmus and kwashiorkor. Thank you very much.